Okay, so good afternoon, everyone. Hope you've all had a good summer. Welcome to the first divisional seminar of the academic year. So today it's a pleasure to welcome Dr. Kevin Mitchell. Kevin is Associate Professor of Genetics and Neuroscience at Trinity College in Dublin. His group are interested in the genetics of brain wiring and its relevance to the variation in human faculties, psychiatric disease, and perceptual conditions like synesthesia. Kevin's current research focuses on the biology of agency and the nature of the genetics and neural information underlying that. He's the author of a book called Innate, How the Wiring of Our Brain Shapes Who We Are. He authors the Wiring the Brain blog and tweets using the handle Wiring the Brain. He's currently writing a second book, Agents, How Life Evolved the Power to Choose. And he tells me that that should be out by next Christmas, so put it on your Christmas list for 2023. Kevin's talk today is what have we learned from psychiatric genetics, which should be relevant to most people within the division, I think. If anyone's got any questions, then please stick them in the Q&A box and I'll address them to Kevin at the end of his talk. So thanks for coming to talk to us today, Kevin.
Super. Thanks, William, for the invitation, and it's really my pleasure to talk to your group and all the people there who are doing such great work in this field. And actually, I feel a little bit like a fraud because I'm not going to really, give a lot of, or maybe any primary data. Really, this is more a conceptual kind of an overview to think, from all the work that people like many of you have been doing in psychiatric genetics. Where are we, and what have we learned about psychiatric disorders, and really what does that information tell us about how we should think about psychiatric disorders? So what kinds of things are our diagnostic categories? What's the architecture genetically of those conditions? What's the relationship between risk genotypes and clinical phenotypes, and what's the right way to think about that relationship and how it's realized in individuals? And ultimately, what will that knowledge get us? So, if we think about psychiatric disorders as diagnostic categories, they're diagnoses of exclusion, really. They're based on clusters of behavioral symptoms, clusters, that recur frequently enough to be recognized as a thing, but they aren't defined by their etiology necessarily. So it's not, or in cases where they are, like say if it's a psychosis caused by syphilis or something, then that's an exclusion criterion for a diagnosis of schizophrenia. So really they're placeholders. They're not necessarily natural kinds, real things that exist in the world, as opposed to just labels that we put on things. And I think that's one of the questions that I want to discuss a little bit and ask whether genetics has anything to tell us about that picture. Of course, they're clinically heterogeneous and dynamic, really challenging to treat in many, many ways, as the clinicians among you will know. But they are moderately to highly heritable. So one question in the field, and really a goal, was that genetics might bring us some clarity on these questions of what sort of things we're actually dealing with. And generally speaking, the goals of genetics across all fields, but in this field in particular, have been to identify risk genes in the hope of revealing the underlying biology, and then translating that understanding to the clinic possibly in many different ways. First of all, just getting a genetic diagnosis, potentially, that leading to more personalized medicine in terms of prognosis and management, and expectations of clinical course and so on, but also ultimately to an understanding of the molecular basis of these conditions that would allow us to generate new therapeutics, which are obviously sorely needed. So what I want to do is go through, where we are now, that many, many risk genes have been identified, and ask whether they have revealed the underlying biology, or what about it, and how that might affect these efforts to translate that understanding to clinical advances. I'm going to talk about neurodevelopmental disorders in particular, so I'm not going to talk about neurodegenerative psychiatric disorders like Alzheimer's or Parkinson's or other late onset things. I'm going to talk about neurodevelopmental ones, and very much bring a neurodevelopmental perspective to the question in terms of how we should understand how genotypes relate to phenotypes. So broadly speaking, neurodevelopmental disorders have been thought of in sort of two groups. There's the rare syndromes like Fragile X syndrome, Rett syndrome, Down syndrome, and so on, which have been known, some of them for 100 years or more, and which are caused by individual genetic lesions, not necessarily to a single gene. Sometimes they are, but it could be an extra chromosome, it could be a deletion of part of the chromosome or duplication or so on. But in each case, it's one genetic insult. And then there's this big group, the idiopathic group, where the causes are not known. And we use just broad terms like intellectual disability or epilepsy, autism, schizophrenia, and so on. And those have been thought of as complex disorders. So the rare ones have Mendelian inheritance. They're caused by mutation or by inheritance of one genetic element, whereas the common classes have been thought of as having complex inheritance. So they're heritable, but they don't segregate in a clear sort of Mendelian fashion. And generally speaking, I guess over the last 20 years or so, there have been two kind of camps in terms of thinking about the genetic architecture of these diseases. There's been the multiple rare variants camp, where the idea is that, like with intellectual disability, each case might be caused by a single rare variant, but they could be in different genes in different families. So the disorder, something like autism or schizophrenia, is genetically heterogeneous. And what's been seen as an alternative, I think, is the common disease, common variants idea, where instead of that, each case would be caused by the inheritance of multiple common variants or polymorphisms that segregate in the population, each with a tiny influence on risk, but collectively might be enough in some people to cross a threshold of genetic burden and therefore induce pathology. And that model then would be that the disorder is polygenic. And I mean, not surprisingly, I think the answer is both, right? So both of these mechanisms are at play. And I think, but rather than thinking that some cases are caused by rare variants and other cases are caused by polygenic burden, my own view is that those things are actually combined, and that most, maybe all cases involve at least one major mutation, maybe several, rare mutations that confer appreciable risk by themselves, but there are real and strong modifying effects of the genetic background. So unfortunately, that makes the most complicated picture that you can have, the worst of both worlds. You have both genetic heterogeneity and polygenicity, which really is a challenge for understanding the biology, understanding how phenotypes emerge, and thinking about how to translate that clinically. So thanks to the work of many people across the field, including many people in Cardiff, of course, that lots of rare variants have been discovered over the years, and those include copy number variants, deletions, duplications, and so on. Now, a growing number of point mutations, thanks to whole genome or whole exome sequencing in very large samples. And the lessons really there are, first of all, that risk overlaps diagnostic categories. It's very rare to find a mutation that specifically predisposes to, say, autism or just to schizophrenia or just to ADHD without increasing risk across those boundaries. These mutations can be de novo or inherited. Of course, the de novo ones tend to be more severe than the inherited ones, and all of them are under strong negative selection. So, which is an important factor, I think, to keep in mind in terms of the genetic architecture, and I'll come back to that a little bit later. So this is just a schematic. Actually, I'm embarrassed to say I forget which paper it's from, showing some of the early discoveries of things like 22q11 deletion, 15q13.3, and so on, which were found to manifest in different patient groups. So studies in patients with schizophrenia or epilepsy or autism turned up these same deletions over and over again. And eventually wider population studies, showed that these are often carried by people in the general population, either with, often with no clinical presentation, but there can be phenotypic effects, for example, on cognitive function, as shown by George Kiroff and colleagues. Exome sequencing has sort of found the same thing when the various genes have shown up in exome sequencing or whole genome sequencing more recently. They pretty much always overlap these diagnostic categories and contribute to risk across those categories. This is a recent paper from Singh et al. Finding some rare variants that are high risk for schizophrenia. So as well as the copy number variants that are shown there mainly in green. So what this picture shows is I think that over time, we look at the top left here, we have this big pool of idiopathic cases. And around the year 2000, we knew of from some cytogenetics or old linkage that there were some rare syndromes that could manifest with those symptoms. Fragile X, Rett syndrome, VCFS, and so on. Over time, as comparative genomic hybridization arrays came into play, more and more copy number variants were discovered. So the pool of idiopathic cases shrinks a bit, and more of them can be ascribed a high-risk variant. And now as we go to exome sequencing and whole genome sequencing, again, the pool of idiopathic cases is getting smaller and smaller, and the number of high-risk single gene mutations is increasing. So that leads to a view that that actually maybe these common disorders are really just umbrella terms. Really, they just hide a lot of genetic heterogeneity that we just didn't know about, because unlike these top, the syndromes at the top here, like Fragile X and Down syndrome and Williams syndrome, which have things like, characteristic facial features and other non-psychiatric symptoms to distinguish them, most of the rare mutations don't. So without doing the genetics, there was no way to tell that these different cases, say, of autism, actually were caused by mutations in different genes. Now, that's a simplistic picture, and I'm going to tell you in a moment that it's not quite right. But just as an aside, to think about the biology of these genes, what does that tell us? There's loads and loads of these rare mutations in different genes have been found. Well, what kinds of genes are they? Well, they're enriched for brain expression, especially fetal brain expression, which makes sense, and that's a good, reality check. They're enriched for neuronal expression over, say, glial genes. And broadly speaking, they're enriched for neurodevelopmental processes. And that's a pretty broad, general kind of category. I mean, there are some, things within that, synapse formation, for example, which may be enriched as well. But it's not very specific. And there isn't really any clear convergence on very particular, biochemical pathways or cellular processes. There's just a very sort of general implication of neurodevelopmental insults in some way. So in terms of getting back to the genetic architecture then, if there are these high-risk mutations, which I've just sort of suggested explain these broad groups as umbrella terms, then why don't their inheritance patterns look more Mendelian? They don't. And in many cases, even though the high-risk mutations, the risk that they confer is not as high as the risk that we know from, say, twin concordance, right? So if we look at twin concordance for autism, something like 80%, what that means is that people who have autism inherited, generally speaking, really high genetic risk for developing autism, higher than those mutations by themselves can explain. So there must be some other things in the background that are contributing to genomic risk, even when people have those high-risk mutations. And we can ask too, then why are the phenotypic effects of any specific mutations so variable? And I think the answers to both of those really relate to how the genotypes get expressed as phenotypes. So if we think of other sources of variance, there are three ones that I think are in play, and two of them are very well known, and the third is I think not so well known. So the first is genetic background, which we'll talk about. The second obviously would be environmental variance, and the third is intrinsic developmental variation. So first of all, genetic background. It shouldn't be a surprise that even really high-risk mutations are modified by genetic background, because that's absolutely the norm, even for the most classically Mendelian disorders, like things like cystic fibrosis or Huntington's disease, which are caused by mutations in just one gene. So everyone who has cystic fibrosis has a mutation in the CFTR gene. So, if your phenotype is just the diagnosis cystic fibrosis, then it looks completely penetrant. But actually at a symptom level, it's not completely penetrant. And if you look, for example, at the severity of lung phenotypes, there's a bunch of genes where mutations in those genes act as modifiers, and mutations in different genes modify, say, the gastrointestinal phenotype or others. So that's just a normal sort of pattern that we should expect to see, and no reason why it shouldn't be the case for psychiatric conditions as well. And I think really what we may have is a spectrum of different forms here. So on the left, this sort of mutation 1 here may be very, very high risk mutations, which tend to have a specific phenotypic profile. They're usually de novo because the phenotype is so severe, so they often occur in sporadic cases. And then the mutation 2 and mutation 3, you may have like mutation 2 there, which is high risk, but not as high as the overall genomic risk we expect. And it could be modified by, the polygenic background, or it could be in interaction with another rare mutation or multiple ones to give some epistatic interaction. So, it could be additive or multiplicative increase in risk and a more severe phenotype. And what we might expect for these kinds of scenarios is more variable phenotypes, incomplete segregation, incomplete penetrance, and so on. So, of course, we have, de novo mutations. Each of us, we have rare inherited mutations, and then we have millions of these common inherited mutations, which many people have been looking for associations with in genome-wide association studies. This is the famous Ripke et al. Study for schizophrenia, which found 108 genome-wide significant hits. I don't know what the number is now. It's many hundreds of significant hits, with an implication that there's actually many thousands of risk variants hiding down here, which might be revealed as samples get larger. And I think the polygenic score at that stage explained about 7% of the variance in the liability score for who got the condition. So again, if we look at the spectrum of these polygenic variants that are associated, they have obviously tiny individual effects, which makes sense because they should be selected against. They're collectively, they can make a very substantial contribution to risk, but the individual variants are under negative selection. Again, the risk is shared across diagnostic categories, so that's true for rare and common variants as well. This is a paper that just came out a few weeks ago, just nicely showing the overlap in risk across different conditions. This is a pairwise comparison, for example, of bipolar, risk SNPs and schizophrenia, which are almost completely overlapping. Bipolar and major depression a little bit less, ADHD and depression, ADHD and schizophrenia, and so on. So all consistent with this view of shared risk across diagnostic boundaries. In terms of the biology, there's thousands of genes implicated. Again, they have nervous system expression, but also very, very diverse functions. They don't zero in on dopaminergic signaling or serotonergic signaling or some kind of metabolic pathways or just one kind of synaptic receptors or so on. That might have been the case. We didn't know when this enterprise was begun, but it turns out not to be the case. There really isn't biochemical specificity. And my view really is that the polygenic risk is a very general signal that reflects the general neurodevelopmental robustness. How well can the system, as it develops, channel development into the most viable outcome, which is the neurotypical outcome, even when it has rare mutations to deal with. So, some people may be better able to buffer the effects of those rare mutations than others. So if you think about this idea of some people inheriting maybe one big mutation or several mutations, that the effects of those can be multiplied by or filtered through polygenic burden. And I would put sex effects here as well, to give an overall genomic risk. And this collectively must be sufficient to explain, the really, really high risk that some people are at compared to really, really low risk that most of the population is at. Now, that's not sufficient. These conditions are highly heritable, but they're not completely heritable. And really what's inherited is the risk, not the disorder itself. So there must be some non-genetic sources of variance. And typically, I think people understand non-genetic to imply or mean environmental. And it doesn't have to. It can do, it just doesn't have to. So it turns out, from twin studies, adoption studies, and so on, that the shared family environment makes little contribution to the liability variance. And people have looked hard for other systematic environmental factors and not really found any that explain, much risk either. So there's something we're missing. There's some reason why monozygotic twins who are completely genetically identical, they share their entire genomic risk profile, and even raised in the same family, in the same general social environment, and so on, are not completely concordant for these kinds of conditions. So something else is making twins different from each other. And I think what that is actually development itself. So the risk is inherited, but how the risk plays out depends on how development plays out. And that's just variable. As you run the developmental program, time after time after time, you just don't get the same outcome, not due to anything outside the organism. That's just the fact that development is a noisy process, an incredibly complex process involving interactions of thousands and thousands of genes to build a brain, to wire a brain, to get all the cells differentiated the right way into the right place, the right dendrite shapes, the right connectivity profiles, and so on. So in fact, the genome doesn't have enough information to specify that endpoint. All that it specifies is the developmental rules that in the processes of self-organization will generate an outcome within a viable range. But the specifics are partly left to chance, really. So you don't get the identical results every time you run the program. And we can see that in twins. So monozygotic twins, obviously in humans, are the only way that we can observe this two runs of the program over and over again. So what we see here is twins who at first are strikingly similar, but as you look a little closer, you can see there's some differences in facial morphology that are very subtle, kind of quantitative differences in size or shape of different facial features, and that's true as well for brain structure. So if we look at brain structure between many sets of twins, with identical twins here shown on the left, the average correlation. This is a very crude measure of cortical thickness. What you can see is that some areas of the brain are really highly correlated between the identical twins and less so in fraternal twins. But there are also these many areas of the brain where the correlations are not very good at all, actually. So, and this is true even when the twins are born at early stages. Their brains are already different from each other because development has just gone a slightly different way between them. So if we want to understand the relationship between genotype and phenotype, any particular individual one, we have to realize that development actually adds variation. There's some potential range of phenotypes encoded in the genotype, but only one of them will be realized through the processes of development. And generally speaking, what that means is that you get a kind of a quantitative spread, as I was showing the facial features of the twins, or this measure of cortical thickness in their brains, which, by itself, you wouldn't think could explain dichotomous outcomes, where one of them might end up autistic and the other one not, for example. However, brain development is highly nonlinear. So there's a lot of contingencies in brain development where one process happening at one stage of development, the subsequent ones absolutely rely on that having happened properly. So what you can get is small little changes at one part of development, which cascade through the subsequent steps of development and which can channel development down one route or another. And sometimes those lead to quite dichotomous outcomes. And we can see that even anatomically, for example, in agenesis of the corpus callosum, massive set of fibers connecting the brain hemispheres, that can differ completely between monozygotic twins, where one of them can have a perfectly normal corpus callosum and the other one not formed at all. And it's, the studies in mice to show what's inherited there is a risk of developing no corpus callosum, but if it starts to form and passes a little threshold, then it can form perfectly fine. And I think of things like handedness or conditions like schizophrenia or autism or so on as also dichotomous outcomes, where there's a probability of going one way or the other. That's what you inherit. Whether you actually end up that way or not is is simply the outcome of development. And this is an old picture from Conrad Waddington, which he called the epigenetic landscape, really using epigenetic in that term, like Aristotle's epigenesis, which was the emergence of the organism through time. And the idea was that this little ball signified, in this, Waddington used it mainly for cell states, so cellular states, with the idea that as development proceeds, this landscape sort of determines the state in which a particular cell will end up. So it might end up as a neural cell or a muscle cell or something like that. But it gets channeled into these particular final states. Now, I think you can use the exact same metaphor to think about phenotypic endpoints of the whole organism. This might be left-handed versus right-handed. It might be schizophrenic versus not schizophrenic. And the point here is that there are certain points in development where there's a little bit of noise can push the developing system one way or the other way. Not due to anything outside the organism, it's just chance. But once it starts getting channeled down this trajectory, then all of the feedback loops and interactions of all of the gene regulatory networks and developmental processes and the cascading contingent nature of brain development may channel it down one route versus another into what can end up being quite discrete phenotypic endpoints. So just to sum up then, I think we can think of the overall genomic risk as including rare mutations, polygenic background, and sex, but that will be expressed through this developmental variation. It could end up with no clinical issues, but it may end up with what you broadly call developmental brain dysfunction, which may then manifest in individuals in any of a number of different ways. So having seen that picture, then I think we can ask, what does the genetics actually tell us about the biology? And if we start from the molecular point of view, as I said, there's enrichment for genes involved in neural development, but no convergence on specific pathways. And generally speaking, there's some clues, I think, we look at polygenic components of risk that they typically at least inversely correlate with, say, genetic scores for IQ, even sometimes for educational attainment as a proxy for cognitive function, which suggests to me, and I think to many others, that may be a generic index of a vulnerability to psychopathology. And that something like IQ, in fact, is a fitness indicator, and it shows the developmental robustness of the whole program and how tightly it gets it into an optimal outcome. So thinking about developmental robustness for me is really the key here, and the robustness of the program is itself a genetic trait, but it's a general trait, right? It's not related to any particular gene. All of the genes collectively contribute to the robustness of the pathway. And natural selection, of course, has selected not just for a given phenotype, not just for a genome that will give a given phenotype, but for a genome that can do that robustly, given all of this molecular noise we get in development, given all of the genetic variation that we all have, and variation in any number of environmental parameters. So one way of thinking about that, I think, is that overall integrity of the genomic program is sort of an analog of cognitive reserve, which you can call genomic reserve, and it's degraded by an increasing load of genetic variants, but in a very non-specific kind of a way. So you can have individuals, and the humans, or we can see this much more directly in experimental model organisms, where they have a low mutational load, quite a robust genotype. That means that the range of outcomes is very narrow. It also means that they can buffer the effects of rare mutations. Whereas some individuals may have a more sensitive genotype, they may have a broader range of outcomes and be more susceptible to rare mutations. And I think, just for the record, that I would put males in mammals with an XY sex determination system over here, because they do have a more sensitive genotype. They are more susceptible to the effects of rare mutations, but they also have a broader range of outcomes for all kinds of traits. Males are just more variable than females. And to me, that sort of illustrates the idea here quite well. So getting back to Waddington's landscape, this was the original picture that he drew, and it's the one that I think is quite well known. What's less well known is the picture of what's underneath here. What is it that shapes this landscape? And the idea is that underneath that landscape, you have these different factors that are pulling down on the like a canvas sheet, so the guy ropes and pulleys as he described them, which would be the individual genes that are collectively shaping the landscape, collectively making one phenotype or another more likely. But the phenotypes that arise are a property of the whole system. They're not a property of the individual genes. And what you can have from genotype to genotype is that you're shifting those probabilities. You may deepen a valley here. You may make this a more likely phenotype and that a less likely phenotype, or you may also, just sort of spread out the width of variance here, where it's just more possible. There's a longer time in which the individual might get channeled into this pathway versus that one. So the genotype shapes this landscape, and then how development proceeds is just a sort of a probabilistic interpretation of that landscape. The important thing being that there are not very direct single gene links between one gene and one phenotype, especially for the types of phenotypes we're looking at. So if we look at that question, then the genotype-phenotype relationship, there are some really big mysteries, I think, that have to be grappled with. And one of them we've been talking about: why do mutations in the same gene lead to diverse phenotypes? And there can be genetic background effects. There can be just the outcome of development. But there's a flip question, which is why do mutations in so many different genes lead to similar phenotypes? And really, why do we see those phenotypes at all? Right? Why do we see psychosis and mania and depression and the symptoms of autism? And why do we see them cluster together in that way? We naturally focus on what we do see, but you can just as well ask what we don't see. I mean, there's thousands and thousands of ways you could imagine that the brain could go awry, that cognition could go awry, that could manifest in very, very different ways, but we don't see that, right? We see a very sort of discrete, recognizable clusters of symptoms along very particular dimensions. And the question is, why is that? And of course, it's not just due to genetics. As I mentioned earlier, something like psychosis can be associated with like infections like syphilis or malaria, can be caused by very different drugs with, hitting different pathways, hitting the dopaminergic pathway, cannabinoids, glutamate receptors, serotonin receptors, and so on. Lots of different genetic insults. And broadly speaking, the condition of schizophrenia, the tendency to become psychotic, hundreds of rare variants and thousands of common variants implicated. So the way that I see this is that, something like psychosis or schizophrenia is a common phenotypic endpoint, but it's a property of the perturbed brain. It's not a property of the perturbations themselves, right? There's something that you can do, that the brain is developing a certain way where becoming schizophrenic is sort of one of the options there. It's one of the possible outcomes that can arise. And insults, many kinds of insults, may just increase the probability that the developing brain will get into that in a way that's really non-specific and unfortunately kind of uninformative in terms of the molecular mechanisms of the condition, because they don't have molecular mechanisms, they have neural mechanisms. So in these cases, the kinds of phenotypes that we're looking at, the functions of the encoded gene products just don't relate proximally or synchronously to the symptoms that emerge. For the most part, there are going to be some exceptions to that, where there's, particular protein really is, very directly involved in the symptoms that we're looking at. I think epilepsy is the most obvious. There's going to be a subset of epilepsy cases where that's true. But more generally, for really neurodevelopmental disorders, the pathway that the developing brain takes is a product of the whole system as it develops. So it's really not anything to do with the molecules that were mutated at the time that you actually have the symptoms, because in fact the damage was done potentially decades earlier. So that's really frustrating, but I think it's important to take it on board. The typical logic, obviously, the goal of doing genetic analysis is, when we're looking at genotypes and how they relate to phenotypes, is that we will be able to infer something about the relationship of genes to functions. I mean, that's why we do experimental genetics in model organisms. and what we hope to gain by looking at clinical genetics in humans. And in some cases we can, right? If you look at a condition like microcephaly, for example, where the phenotype that we're looking at is really direct, it's really concrete and measurable, and it's also very proximal to the functions of the genes. So the phenotype is a smaller brain that's caused by impairment of the normal regulation of stem cell division in the cerebral cortex. And when you do the genetics and you find these high-risk mutations by linkage in different families, then what you find is that all of those genes in some way directly, the proteins directly affect the division of stem cells into neurons and so on. So nice concrete phenotype. The genes are really proximally involved in the functions that you would expect. Now, it's really hard to say for psychiatric disorders, well, what cellular functions should we expect to underlie the highest levels of cognition and perception and language and sense of self and social cognition and so on. And it's just not obvious that there is a set of molecular processes that underpin those things because they're the highest level functions of the brain overall. So one way to think about that is to sort of compare these concrete versus what I call emergent properties. So imagine you had a fighter jet or a range of fighter jets, right? A bunch of individual fighter jets that varied slightly. And what you were looking at was turbine function. So here's the turbine. You were looking at turbine function and how that differed between different fighter jets. And what you would expect if you looked at that was that it was differences in these parameters that explained and underpinned differences in turbine function. But if you were looking at something like performance, the overall performance of the whole jet is a sort of a nebulous emergent property. It doesn't depend on just one particular element. It depends on all of them. It depends on their interactions in a really non-linear kind of a way. And so you wouldn't expect to just see turbines showing up underpinning performance. You'd expect to see all kinds of complex variation in all of these parameters that might contribute to that. And so this is the kind of level that I think we're at when we're looking at psychological cognitive functions that are impaired by psychiatric symptoms. So if we ask, what are risk genes for? They're not genes for autism or for schizophrenia. First of all, they're not specific to those conditions, and obviously that's not what they're for in terms of a purpose, right? But they're also not genes for social cognition or for working memory, because those, sometimes people have thought of those as endophenotypes that might be closer to the action of the genes, but those are high level functions of the whole brain. They don't have very proximal molecular underpinnings. So really they're genes for neural development or neural function or plasticity, or more accurately, they're mutations that affect those processes. So what that means is that the pathological states really don't relate to the functions of these genes. They're emergent properties, what you can call in engineering terms, failure modes. And I know that's not a nice term to use to apply to human beings, but it's a term of art in engineering. Failure modes of dynamical neural systems. So any complex dynamical system may be selected for or designed for, if it's an artificial one, some particular outcome, some particular kind of functional range, but they will also show unanticipated modes of failure or variation, which can be quite sort of bizarre and have qualitatively novel properties. In a way, like I think a lot of psychiatric symptoms are qualitatively novel. They're not just a decrement in function. They're a shift to a different kind of regime that I think tells us something about the system. So one way to think about that is as attractor states in dynamical systems, where, for example, if a system moves away from this sort of point that describes a state space or the regime that it's operating in, that most of the deviations can be buffered and it will bring it back into a normal operating range. But you might also have, and that might be our typical outcome of development, but you might also have a maladaptive functional regime, one of these novel qualitatively new regimes that emerges that this typical brain could be pushed into, and that could be more or less likely for different brains. And you could have alternate regimes that emerge here. And in a way, what this means, if this view is right, it actually means that even though the etiology of these conditions is quite overlapping, the risk mutations overlap for autism or schizophrenia or bipolar disorder, these functional outcomes may still be discrete. They may still be natural kinds in terms of particular qualitatively novel regimes that the brain can get itself into. So it's the genetics doesn't necessarily undermine the idea that the symptomatology is pointing to real things. It just says there's not a direct link between those two levels. So just to sum up then, broadly speaking, I think we've learned loads from psychiatric genetics, a huge amount. We've learned that diagnostic categories are not natural kinds ideologically speaking. The genetics is heterogeneous and it's polygenic, which is not really good news for making sense of things, but that seems to be the way it is. And really, there aren't very clear pathways or processes, and the pathology just isn't closely linked to the genetic etiology. There aren't close molecular proximal underpinnings of, thinking that the CIA is tapping your phone, for example. So for me, I think of psychiatric conditions as emergent phenotypes of neural systems, these maladaptive attractor states, and the genetics is just what can push you to be more likely to end up in one of those states. So if we go back to our wish list for doing genetics, we can have a little report card here. So loads and loads of risk genes have been identified through the collective efforts of so many people around the world. They've revealed the underlying biology but what they have revealed is that it's really general, neurodevelopmental. There aren't really specific pathways there, and that obviously makes it difficult to translate the understanding to the clinic in the way that I think people had hoped that they could, by analogy to say cancer, where particular molecular pathways are really closely related to the type of cancer that emerges and might be treatable in a personalized way. I think that these goals here are going to be much harder to realize and take a lot more work. And really, I think what's important to understand is that when we're doing genetics and other sorts of epidemiology, we're down here, right? We're at the etiological point. For some kinds of conditions, that's going to be close to the pathology, but for psychiatric symptoms, it isn't. So we have to go through, to understand pathogenic mechanisms, we have to go through biochemistry and cell biology, and especially developmental biology. And to understand pathophysiology, this is the level at which to look for explanation. So it's the cellular circuit systems level. It's linking neuroscience and behavior. It's no longer genetics or genomics. And especially when we get to psychopathology, we're going to have models, I think, of computational neuroscience and cognitive science and psychology that explain how these effects from here cascade through the development of the nervous system, ultimately manifesting in the kinds of psychological symptoms that we observe. This is a kind of a grand schematic, really aspirational, I think. But to me, it highlights some ways that progress could be made, because I don't want to leave people with a sort of pessimistic outlook. I actually have a really optimistic outlook in terms of how we're going to make progress. It's just that it won't happen from genetics or genomics alone. I think if we have patient cohorts, we can obviously identify disease-causing mutations. That may allow us to subgroup patients. This already allows an ongoing clinical relationship that's informed by genetic diagnosis. But having these rare mutations, it gives us an experimental handle that, frankly, common variants don't. It gives us some experimental purchase where we can model the effects in mice or other animals, and ask the right questions at the right levels. We can ask about the biochemical effects on cellular things. We can ask about deviation in cellular function on circuit level and on systems and so on. And at the systems level, we may be able to map between human functions and behaviors or neural functions and systems that we see in animals, ultimately to get to a point where we can say, I understand the system that's affected here and how, and maybe there's a new therapeutic that could come into play that can correct the emergent systems, as opposed to thinking that our new therapeutics are going to come from the biochemistry of the disease-causing mutations themselves. So I think that's all I wanted to say. I'll leave it there. Thank you very much for your attention, and I'm going to cheekily give a little ad here for The book Innate, which William mentioned, which is supposed to be accessible to a broad audience, and then this other one from 2015 on the genetics of neurodevelopmental disorders, which is much more technical. Thanks very much.
Great stuff. Thanks a lot for that, Kevin. So if anyone's got any questions, if you could stick them in the Q&A box, I'll read them out. So we've got one there from Anthony Isles. So about the interaction between genetics and development. So given that the process of very early development is critically dependent on factors present in the egg at fertilization, is it worth integrating genomic data from both affected offspring and their mother in order to gain a more holistic view of vulnerability?
Yeah, thanks for that question. It's a really interesting one. And, you know, obviously in flies, for example, there's a lot of the screens that were done that led us to understand early development of the fruit fly were looking for maternal effect mutations, because the early embryo in the fruit fly is patterned really by maternal proteins and RNAs that are put into the egg, and mutations in the mother can affect that cellular environment within the egg in a way that leads to dramatic phenotypes in the offspring. In mammals, the zygotic genome gets turned on at the 2-cell stage. So there isn't a long sort of period of maternal instruction of the early developmental processes. That doesn't mean, however, that there aren't mutations that could be in the mother that would affect that cellular environment, at the very early stages that might cascade through the rest of development. It's something that I've asked people who are doing the human genetics, because if that's true, then the clue should be that there should be maternal effect mutations that increase risk of psychiatric illness. And I think people have looked somewhat, I don't know how hard they've looked. Obviously, you need the right experimental design to do that. My feeling is there haven't been many that have been discovered. And I don't know, frankly, whether that means they haven't been looked for hard enough or in the right way, or they don't exist. So my feeling is it's an empirically open question as to whether we should expect those kinds of things to exist and risk, to be that kind of risk to be there. And of course, we know there are environmental factors, like that cause like fetal alcohol syndrome, for example, that manifest in effects on the developing fetus in ways that ultimately lead to expression of intellectual disability and autism, anxiety, and ADHD, and so on. So it's perfectly plausible that there could be genetic factors as well that affect somehow the environment of within the egg, where they wouldn't even have to be passed. The mutations themselves wouldn't even have to be passed to the offspring to have an effect. But yeah, so I just don't know where the state of the art is now and how hard people have looked for those kinds of things.
Okay, thanks, Kevin. So we've got a second question from Howell Williams. Says, very interesting talk, thank you. So he agrees with your ideas about how stochastic changes can alter neurodevelopmental programs and lead to different endpoints and phenotypes. His personal view is that regulation of genes during neurodevelopment, spatially and temporally, can potentially be a rich area of research, and that genetic variants within these regulatory regions will have a range of effect sizes from small, for example, those shown by GWAS and polygenic analyses, to large effects, so known genes with point mutations, for example. Does that view fit with your neural mechanism?
Yeah, absolutely. And I mean, a lot of the genes that have been found with high-risk mutations, in particular in autism, for example, are chromatin regulation genes or transcription factors, all of which are doing the job of regulating genes during development. So it absolutely makes sense that mutations affecting the regulation of genes will shift those probabilities. The challenge, I guess, is how we should conceptualize that, right? Should we look for one particular mutation or one particular gene whose job it is to, make this kind of cell type or something like that? And what I was trying to get across, I guess, is the idea that development is really a collective process of the gene regulatory networks across the whole genome. And of course, you can find, specific effects of particular mutations on particular phenotypes, but even there, those proteins are often involved in other gene regulatory networks in other cells and other tissues at other times of development. There may be this particular pathway may be particularly sensitive to one or other. But I think you can get sucked into the idea of expecting more specificity between individual genes and individual phenotypic steps or processes of development. And to me, really, it's a collective picture. What the genome does is generate effectively an energy landscape that the developing system then follows. And so unfortunately, that means that our reductive methods of gene discovery, which can be really powerful, are not easily translated into a reductive understanding of the developmental processes, because the processes are not linear and specific to those genes. Right? So I think it's right to think about developmental trajectories, but I think we should be taking a systems biology kind of approach to that. And thankfully, with the tools that we have at hand now, where we can analyze the expression of all the genes in the genome, at a single cell level across many, many stages of development, if you have, say, access to the right tissues, obviously you can in model organisms, then I think we can have a conceptual framework that can still be data driven. So just because it's holistic doesn't mean it's kind of hand wavy and nebulous. It means we just can measure all the genes at once and think about the shape of the landscapes that emerge that will channel development in one way or another.
Okay, thanks, Kevin. So third question from Mick O'Donovan. Can you clarify what you mean by identifying a specific pathway? Schizophrenia, rare coding variants, CNVs, common variants all highlight synaptic function at the top of the list. That is, of course, still pretty non-specific and pretty general. Yeah. So can you define what it is you want to see that might change your mind that there's a link between genetics and pathophysiology? So what results from genome analyses would allow refutation of your hypothesis?
Well, that's a good question. I mean, looking into, at the start of this long process, there were hypotheses about, say, dopaminergic systems being affected in schizophrenia, serotonergic systems being affected in depression, and so on. If it had turned out that those pathways really were, there really was convergence on those, that the genetic lesions were concentrated within the genes encoding proteins doing those functions, well, then that obviously, would have been a result that proved that kind of specificity. As it happens, if you look statistically speaking and you think of these systems as interconnected, just statistically speaking, there are many, many more ways to affect the functions of the serotonergic pathway in the brain than just the genes involved in serotonin metabolism and so on. Like, you can mutate all kinds of other genes that have indirect effects on that. So even though their effects might not be as large individually, there are so many thousands more of them that the genetic architecture of that trait will be dominated by these indirect effects. And for example, Jonathan Pritchard has shown that really nicely with the most proximal molecular effects you can think of, right? The most proximal phenotype you can think of, which is just the expression level of several proteins. And when you look at that, you can find there are some mutations in the regulatory domains of those proteins or transcription factors that very directly regulate them that contribute to genetic variation in that trait. But there are also thousands of other genetic variants where there's no sort of logical association that you can draw or proximal link between them. And those thousands of other genetic variants actually dominate the percentage of variants in the phenotype. So even with the most direct proximal phenotype you can imagine, the genetic architecture is dominated by indirect effects. And so for me, the fact that that's true for the highest level cognitive functions that we have is not a surprise. And so, I mean, what would refute it? I don't know. It would have been refuted, I think, if there were really specific pathways there. I guess the question, operationally could be, well, what do you do now? Is it worth, for example, doing more and more genome-wide association studies on a larger scale with the hope of finding more specific biology? If that's the goal, then I would say no. I would say that the studies that have been done so far indicate that there isn't specific biology to be found there. That's my view. And it didn't have to turn out that way, but that's the way I interpret the data to hand so far. And so it just means we're going to have to look for specificity at the emergent neural level, not necessarily at the proximal molecular level.
Okay, thanks. So fourth question from Mike Owen. Many psychiatric traits are pseudo-categorical in that they appear to represent extremes of more general variations in behavior which share genetic risks, for example, autism and autism spectrum. So what is called a disorder is to some extent arbitrary. Maybe the canalization model is a bit simplistic, or maybe Mike's understood.
Yeah. Well, so I mean, the canalization model definitely is simplistic, right? I mean, in a sense, that's partly what's good about it, but absolutely, of course, it's simplistic and very, very abstract. Right? So the sort of the idea of psychiatric disorders just being the end of a continuum is interesting, and in some way, the genetic overlap with general traits in the population supports that view, but not completely, I think. Clearly, there can be, individual rare mutations which increase risk of these conditions very discreetly. And it's not just that the individuals are at the sort of extreme end of several different dimensions of behavior at the same time. The fact that these things cluster, right? I mean, that we see impairments of social cognition and, repetitive interests, for example, in autism and the other conditions, symptoms of autism as a recognizable cluster, more or less, suggests to me that there are some sort of, like I said, endpoints or regimes of endpoints of development or regimes of neural activity that represent those states collectively with a cluster of symptoms. So I don't know what the dimensionality sort of aspect implies going forward. I think there was a hope that that studying the genetics of, say, social cognition across the typical range would inform on the genetics of autism, or you could do the same with genetics of working memory and schizophrenia or whatever. I mean, Mike probably knows better than I do now what the current state of play on that is. To me, it's kind of sort of true, but not completely true. I guess I tend to think of it in a way, again, as this sort of combination of things where you can have a polygenic range of variation in these dimensions, and then some insults that have, can push the brain into an altered regime that affects those dimensions, and maybe there's an interplay between those. I know that was really hand-wavy response. Sorry for that.
Okay, and I was just going to finish with a question from myself. So another way you might get variation between individuals is via somatic mutation. So to what extent do you think that's playing a role, and how might that interact with this developmental program?
Yeah, it's a great question, and many people have raised the idea that somatic mutation could affect things. My own view is that somatic mutation will be so diverse across the developing organism, including the developing brain, that you get individual neurons here or there, or maybe clonal patches of neurons that relate to that all carry a mutation that arose somatically. Unless it's a mutation that arises really, really early that affects most of the brain, then I don't think it's going to contribute to the phenotype that much, or at least I don't think there's good empirical evidence that mechanism is at play for these kinds of conditions. It's completely possible that it could be. I just haven't seen the evidence that it is, except for conditions like ones where there's some overgrowth, cortical malformations, for example, where the mutation itself gives the cells that carry that mutation a growth advantage in the brain, and thereby they come to dominate the landscape there. So unless something like that is at play, it's hard for me to see how sort of scattered individual mutations would affect the dynamics of the whole system. But it's perfectly plausible and really difficult to rule out.
Mm-hmm. Okay. I don't think there's any other questions. So with that, I think we'll wrap it up. So thanks again for joining us, Kevin, and thanks for a great talk.
Okay.
Thank you everybody for coming, being in the audience.
Brilliant. Thanks very much, and thanks everyone for coming.
I'll send you a copy of the talk, Kevin. Okay.
Super. Thanks a lot.
Right, thanks everyone. Bye.
Bye all.